Good morning. My name is Jeannie Montblanc, and on behalf of the Great Basin Fire Science Exchange team and our partners, I would like to welcome you to this webinar titled Wildland Seed Collection and Extraction, presented by Kayla Herriman and Sarah Garvin from the Forest Service Region 6 Bend Seed Extractory. Before I introduce our speakers, I want to let you know that you may ask questions of the speakers or me at any time during the webinar by typing them into the questions pane of your control panel at the top right of your screen. I will keep questions for the presenters in the queue and field them after the presentation. I also want to let you know that whatever you do in your control panel does not affect the webinar presentation, so you're welcome to type a test message or test your audio at any time during the webinar. If you're having problems with your audio, please open your audio pane and check your audio options selections. Now I would like to introduce our presenters. Kayla Herriman is the Seed Extractory Manager for the Forest Service Region 6 Extractory in Bend, Oregon. Kayla has worked for the Forest Service and BLM focused on plant regeneration for over 10 years. Her experience is more conifer-oriented in regards to cone collection, nursery work, and outplanting. As a research assistant at the University of Idaho, she focused on sagebrush and then moved into native seed. Sarah Garvin is the Bend Seed Extractory Assistant Manager. Sarah has also worked for the Forest Service and BLM for over 10 years, with a focus in botany. Her experience ranges from field surveys, seed collection, and seed processing. Sarah has also worked at a native plant nursery as a greenhouse manager and is very knowledgeable about native species propagation. Welcome, Kayla and Sarah, and thank you for being here today. Okay, I'm going to make you the presenters. Oops.
All right, can you see our screen now?
Not yet. There we go.
Okay.
And I can see your icons at the bottom, so if you want to do the screen clean option, it'll remove those. I mean, it's not that big of a deal, but if you prefer.
Oh.
There we go.
So I'm Kayla, and I just wanted to welcome everybody to the second webinar in the series. As you guys know, it's called The Right Seed in the Right Place at the Right Time, Tools for Sustainable Restoration. Our goal today is to focus on seed collection and processing, focused on wildland restoration. So we're going to talk a lot about small collections and what you might do out in the field in terms of collecting seed. So the first thing we want to talk about is why this is important. In terms of collections, restoration is really a priority for natural resource management, and it seems like a lot of our organizations are focused on using native seed, which is really great because native seed fosters diverse populations, strong populations, and those that are well adapted to the local environment. So by collecting native seed, you have an opportunity to impact those populations in both of those avenues. So it's really important that we're moving in the right direction in terms of restoration and making sure that we're creating the strongest populations into the future. So talking about seed needs and planting, this cycle for native seed is important because we can see that each step is connected to the one either prior to it or the one after it. This is really important for us because if we don't understand what it takes to collect seed, that can hinder how we process it here at the extractory. But knowing how it's processed can also aid seed collection. So it's really important to think about this, this graph here and know that all these steps contribute to the end product, and knowing the quality at each of these steps is really important, because it really contributes to a quality end product. So if we can each do the best we can at our stage, then the result is going to be just that much better. So what are we going to discuss? We're going to talk a little bit about planning for seed collection with some tips and guidance, how to implement seed collection, some different tools and methods and best practices that we've found, seed cleaning and testing, some of the benefits and challenges, and then different options for those, and then a little bit about storage and shipment and how important that is to not only your seed, but potentially your project that you're working on. So in terms of planning, some of the basics that are really important to consider are why am I doing this? The purpose and need. Because planning for seed collection is really as important as what you're going to do with your seed. It's important to understand all the factors that may affect your seed and how you'll use it. And knowing what you will do with the seed can also determine maybe how much you're going to need, or potentially understanding how much each of those steps is going to cost. You don't want to plan on large collections that cannot be processed and may result in a loss of viability. So really thinking about how am I going to use the seed, what do I need it for, and understanding the time frames, the budget, the amount of work that goes into collecting it, and who might be able to be available during those key times of seed collection, and how you're going to use it, really all contributes to what you're going to need to plan ahead of time before collection actually happens. And then location, thinking about the location. It's really important to be familiar with your species range and the local ecology. It will benefit the logistics. So looking at seed transfer zones, how accessible is that population, and then how suitable is that population. So really thinking about your seed location before you collect is important. And then estimating what you're going to collect in terms of quantity and quality, and then documentation. So keeping in mind all that stuff, I know it's a lot, but it's really the important basics to focus on before you do any seed collection. So the first one on there, what is the purpose for seed? So some things to consider. What is your seed going to be used for? There's lots of different reasons that people collect native seed. Restoration, genetic preservation, research. So the why of the collection is really important. And then what is the project time frame? Are you going to use the seed right after collection? Are you going to use it in the next season? Is your project a few years out? Thinking about project time frames and how the project can get accomplished to meet those goals can determine what your availability is for seed. Potentially, if your time frame is a few years out, you might be able to collect over a few different seasons and then combine your seed together. And then, do I have adequate funding? This is a question I think we ask for every project. But it's really important with seed because all of the aspects, once they come together, sometimes it's surprising how much it can cost to really go out and collect seed, have your seed processed, have your seed shipped, have your seed stored. You know, from the time you collect it to the time you get it to either the field or a nursery, there's a lot of factors that can come into play. So really knowing what it takes to have a seed collection go from the initial planning stage all the way to the final stage, making sure that you have adequate funding for that to consider all those aspects. So who's going to collect the seed? It can be done by individuals or groups. We have experienced a lot of different folks from all over the country. It can really be done by anyone, but the most important thing is that they are trained and that they understand what the goal is. It's really important that they understand species identification and a little bit about seed biology, because that will contribute to a successful collection. So what about seed production? There's a few options for production if your field collection is not enough. And we experience this fairly often. Contracts will be discussed in the next few webinars, and purchase of seed will be discussed in the next few webinars, so I encourage you, if you're interested, to listen to those. But it's really important to understand that if you're going to have your seed increased, that however you're doing it, whether it's a contract or a task order, to make sure that a lot of the important information is in there so that you are getting high quality seed and that it meets the specifications you need for your project. So there's federal nurseries that will do seed increase, like J. Herbert Stone. There's also a 5-year IDIQ commercial services contract that includes not only some collection of native seed, but it does include increase in seed production. So I encourage you to check those out if that's something you're interested in. So moving on to where to collect. This is important to consider because of seed transfer zones. Collecting from a wide genetic base will foster a more diverse gene pool for your outplanting site, making your project more successful. This practice can protect a planting against unforeseen biological and environmental stresses, and it also helps prevent inbreeding. So thinking about transfer zones, accessibility, population suitability, all of those things can help contribute to a good location. So in talking about location, what are the benefits of transfer zones? There was a webinar previous to us about the use of seed zones to ensure adapted plant material. So if you want to know a lot more about them, please watch that webinar. But it's important to know in terms of collection that increased supply and use of native seed will influence the capacity of your restoration, and you have improved restoration success, and it also facilitates the sharing and exchange of seed because these zones can cross land ownerships, administrative boundaries. It allows us to share seed in a way that we may not have before. So if you are collecting based on seed transfer zones, it allows seed to potentially be used for a lot of different projects. So taking into account that is really important. So Sarah is going to talk more about what makes a suitable population and evaluating the population, and a little bit more about actual collection.
Thanks, Kayla. So the question's going to come up, and we get this question a lot at the extractory. How do I know where to collect? And Kayla's already talked about seed transfer zones. Once you've kind of narrowed your area down in that regard, you'll try to zone it into a specific population that you're going to target. First, you might want to consider whether you can get to that population or not. Often we hear about high elevation species that people want to collect. Just be aware, it's really hard to get to some of those locations even once, not to mention 2 or 3 times that a collection really should be made across. So how long does it take you to get there? The cost of getting there, is it 20 miles away? Is it 50 miles away? How many people are you going to need to hire to make the most of that trip out there? and just realize that you're going to need to make multiple visits to capture the phenology of that population. Also, while you're there, you want to evaluate whether there are enough plants at the population. Maybe you need 2 pounds of material, and you know that going into it. It's helpful to visit the population early in the stages of planning and know if that's even a potential with the number of plants on site. You want to be able to collect from the entire population and to ensure the diversity, you're capturing the diversity on that site and that it's going to meet your project needs. So on to the next slide. You want to create a plan that allows for equal and random samples across the population. You don't want to show up, see that you have enough plants in a tiny little corner of the population to meet your needs and just grab from those plants. You want to know the extent of that population and grab from every portion of that population. Pay particular attention to microsites. Those are where you're going to get diversity, and be sure to sample from those and create a plan that allows you to sample from those. Again, sample from the population throughout its dispersal season. Every species has early, mid, and late blooms, and make a plan to visit those early in the season, in the middle, and at the end to get a bit of seed from each one of those stages. Going on to the next slide. This is a question we get a lot at the seed extractory, is particularly from people who are either new to collecting or who are collecting a species that they're not that familiar with. So they often ask, you know I have to produce 10,000 PLS. How much do I need to collect in the field to meet that need? And determining the amount of seed you need to collect definitely starts with knowing how the seed will be used. Is it going to be used in direct seeding, maybe a hydro-seeding project? Is it going to go to a nursery for seed increase? Is it going to go to a nursery for grow out, container grow out, or bare root grow out? Or is it going to go into long-term storage? At the very minimum, you need to know that before you collect. You might be able to, in terms of like direct grow out, you might be able to do some research online and get good information about the number of pounds of seed needed for your species per acre. Also, you should be able to contact nursery staff, and they could let you know either the number of seed you need or the number of pounds of seed you need to meet increased projects, container grow out, or bare root grow out targets. Also, maybe take into account at this stage that there's some species, if you're intending to store them, some species that just do not store at all. So make plans to account for that. Maybe collect more than you typically would for those species. At every stage in the planning process, I'd really recommend learning as much as you can about that particular species if you're not familiar with it, particularly if you haven't collected it before.
Sarah,
so on the next slide.
Sarah, can I jump in a second? What's PLS?
PLS is an acronym for Pure Live Seed. Okay. And it's just a way for, it's just an indication of how many seed in a clean product, how much of that seed is viable.
Great, thank you so much.
Determine it. Yeah, that. So on this slide, you might wonder, how in the world am I going to make an estimation of how much to collect in the field? It's not an easy thing to do. It's the amount of seed you're going to get out of the collection varies so much by species. We get questions all the time, and so here at the Seed Extractory, we've processed over 13,000 wildland collections, and we kind of feel like we're at the stage where we can give some good information back to the public about yields from those collections, particularly for the species that are collected most commonly. So like the slide says, we have yield data available for many commonly collected species. We also have average seed weight is known for many commonly collected species. And with the two of those numbers, you can actually come up with a pretty good estimate of what you need to collect in the field. And you might have this disclaimer down at the bottom of this slide that says yield data is meant to be used as a guide and not a guarantee, and that's really important. You know, don't take an estimation and expect that that's exactly what you're going to get in the field. It's just a number to go from. Of course, we assume that thorough planning has gone into your collection in regards to location and timing of collection, and that's always going to be how you make a great collection, is the planning that goes into it. So we put together 3 scenarios to show how different species and uses might affect the amount of raw material you need to collect in the field. This first scenario talks about Wyoming big sage, and we get a lot of collections of Wyoming big sage here at the Seed Extractory, so we thought this was a good slide to include. In this particular scenario, the client needs 1.5 million seedlings for a transplant on a project, and they plan to do a burial root grow out, and have been told by the nursery that they need at least 2 pounds of seed to meet the targets. I'll just point out that I have no idea if these numbers are accurate. I just kind of threw some out there, so you don't go by these numbers. But so average yield data from the Bend Seed Extractory shows that wildland Wyoming big sage collections typically produce 4% of their weight in seed, not very much. So if you divide 2 pounds by 0.04, you get 50 pounds of raw material is needed at the collection site, which is a lot of seed. And at this point, once you know this number, you might have to go back and reevaluate how many collectors you need, or whether your population is going to produce that amount. Next slide is a scenario where snowberry is needed for a project, and the client has requested that 1,000 plants be grown in containers, and the nursery is asked for the client to provide them with 3,000 seeds to meet that target. From our data, we know that the average seed per pound for snowberry is 66,700 seed. So if you divide 3,000 by that number, you get 0.0449 pounds of clean seed. That's clean seed. You still don't know how much you need to collect in the field. So the average yield for snowberry is approximately 4%, similar to sagebrush. So divide the pounds needed by the yield, and you get 1.12 pounds of raw material of berries that you need to collect in the field. Berries are particularly difficult to estimate by doing cut tests in the field. To me, they're just hard to estimate how much to collect. I would say, other than a few species that we typically have low fill, we don't get enough raw material from berries as any other group. So the next scenario is for Atriplex canescens, and I was thinking of the Seeds of Success program here in particular. And I chose Atriplex because it's one we struggle with. So for the Seeds of Success program, 10,000 pure live seed are required for long-term storage. Often a client collects additional material with the intention of having it grown out in a nursery. So in this scenario, they really need 15,000 PLS, pure live seed, which is different than just seed. So our average yield data show that about 7,700 PLS per pound of raw weight can be expected for Atriplex canescens. So divide those two numbers and you get that just under 2 pounds of raw material are needed in order to meet that target. Atroplex is an incredibly hard species to collect and even harder for us to process at the extractory. It's one of those species that we can't remove the seed from the utricle. So our end result, the finished product usually has a low fill. We just can't get it down to pure live seed the way we can for other species. So I wanted to include Atroplex in one of these discussions because it is so different. And this scenario is typical for other Atroplex species, many conifers, and many species producing fleshy fruit. We're just not able to extract them to the desired purity or fill, and makes it harder to meet targets. If you're unsure whether your species is like that, I would suggest going through and making an estimation using different methods for many of these scenarios and taking the highest number out in the field with you. So, yeah. So we've put together a list of 45 species where we have that are most commonly collected, that we have received the most collections here at the extractory from. And we divide them into 3 categories: forbs, grasses, and shrubs. And if you look at the table, you've got for each genus and for each species, we have an average yield percent, an average PLS per pound of raw weight, an average seed per pound, and the number of records that were used to generate those numbers. So the average yield really is just the clean seed weight divided by the raw weight. It doesn't account for PLS at all. So when people see these lists, I anticipate that there'll be a lot of questions of, well, what do I do if my species isn't on the list? And that's probably going to be the case. There's only 45 there, and we've cleaned over 2,000 species. But please feel free to call us or email us and ask us if your species is one that we have cleaned, and we would be more than happy to give you any data that we have on your species, whether it's only been collected twice and cleaned twice, or if it's been collected 100 times. I think any information helps. So on to the next slide should be a table similar to this one, but it is just a list of grasses. The only thing I might point out here is if you look down to the Hesperus stipicomata column, that's particularly interesting to me. It's the heaviest grass seed that we process here at the extractory. The average seed per pound is around 75,000. So you might expect that it would have a really good yield just because the seed is so heavy. But if you look over the yield, the average yield for over 100 collections that we've processed is only 16% compared to some other species, some of the elmises and fescues. I might also point out that if you look at Discampsia and Spirobolus, the average seed per pound for those two are actually very similar. And if you've collected them, you know they're good producers. And the average yield is actually very similar to 28% for Spirobolus, 32% for Discampsia. On to the next slide. This is our shrub table. And you might notice right off the bat that the average yields are significantly lower across all these species. The one thing I might point out here is the Atroplex, those 3 Atroplex species. The first 2, Canescens and Convertifolia, have really high average yield, 28 and 26%. But that is not taking into account the fact that we can't process those to a high fill percent typically, I mean, it's not unusual for either one of those species, the finished product to be below 50% fill. So that yield percent there is not a very good indication of what you're going to get on your project in terms of germinating seed. Whereas Atriplex polycarpa, we can actually get the seed out of the utricle without damaging it. So the yield looks really small, but if you look over into the average PLS per pound of raw weight, it's much higher than the other two Atriplex species. Okay, at this point you're probably saying, I have to be ready to collect. I've done a lot of planning. Surely I'm ready to collect. Before you go out anticipating being able to collect, you want to visit your site, examine a representative sample of the seeds, not a huge amount, just a representative sample across the population. Estimate the amount of empty and damaged seeds. It could be that there are no filled seeds at that site, that they're never going to produce anything other than empty and damaged seeds. Hopefully the majority of the seeds are mature and fully formed and you can continue on with your plans for collecting. I might just mention here a few things to think about. Avoid collecting from single individuals or from small populations. Collect from the entire population because you want to get the diversity. And ensure, or as best as you can, try to collect seed from similar habitats as your project site. I'm thinking here of like maybe Oregon sunshine. You don't want to be collecting it, if you're in the high desert, you don't want to be collecting it on the west slope of the Cascades and planning to use it out as you're on your project. Take into account elevation, aspect, and soils, as well as seed transfer zones. And maybe just a bit about ethics. Seed collecting ethics. Try to stick within collecting 20 to 30% of the seed available at that site. Once you get over 50%, you're having a significant impact on that site. Avoid soil disturbance, and maybe avoid weedy sites. If you're trying to collect squirrel tail from a site infested with medusa head, just know that that's never going to be able to get out of your finished squirrel tail population. Those two seeds are too similar in size and weight. We'll never be able to use it, and chances are a nursery will not accept that seed. So, also at this time, you should be assessing whether your seed is mature. This can be pretty intuitive. The slide on the right, arnica, that seed is as mature as it's going to get. It looks to me like if you touch the stem, the seed would fall right off the flower head. Not always so easy. The slide on the left is Oregon sunshine, and if you've collected Oregon sunshine, you know that it almost never falls freely from the seed head. You almost always have to rub it out. But I can tell that the majority of seed in that collection are mature just in terms of color and the way that the flower head looks. The flower's almost gone. On to the next slide. Cut tests are going to be necessary for a lot of species. For sure, fleshy fruit, I'd recommend it. Cones, anything in a cone. At this point, you're looking for maturity as well as empty seed or damaged seed. Even if you feel like you're like in the arnica, for instance, if you feel like that seed is mature and you know that it's probably not going to get any better than that, I would still recommend doing a cut test on a portion of those seed to determine whether any of them are even filled. And also, don't wait until collection day to do your cut test. You might you might not know, or you might not know what that seed looks like when it's mature if you haven't cut it when it's immature. It's nice to have the two comparisons. On the next slide is a good example of how color can be used to assess maturity. This thimbleberry, the fruit on top is obviously immature. The one that's squished in the hand is mature. Berries are hard. I mean, they're pulpy, they're juicy. It's hard to do cut tests. This is probably a better method for most berries. And I'll just mention here, ripening does not occur after collection. So if you collect that berry on top, hoping that it's going to ripen in the bag, it's not going to happen. Oh, this is a good example of how often it's easy for species in a pod or a capsule. If that capsule or pod is starting to split, if it's dry, if the stem is dry, there's a really good chance that that seed is mature. I'm thinking of astragalus in particular. You really don't want to collect something like that unless it's the pod has started to split. That seed really needs to be ready when you're collecting it. And I'll just mention, here's a good time to really take the time to research your species, because some plants progress very quickly. They're mature in the pod one day, the next day the pod opens, and by the end of the day there's no seed left in that pod. Lupin, poppies, cleome, fireweed, milkweed, once that pod, it starts to dehisce, those seed are going to be lost quickly. So chances are you probably already know that about your species, but research never hurts. And likely that you're going to have to make more visits to populations that have those characteristics. So going on to the next slide, finally you're ready to collect your seed. And these two photos are a great example of how we at the Seed Extractory like to see seed collected. On the left, the Poa secunda she's stripping the seed from the stem instead of cutting the stem. And the person on the right is pruning off flower or seeds from the tips of the branches instead of cutting off a whole branch. For us, we have a hard time processing anything with stems over 6 inches just because our machines are designed for mechanically harvested material. So we have to go to the extra step of putting it in a different machine just to get things off of the stem before we start to extract it and finish it. And the more machines that need to be used, the more likely you are to either damage seed or lose seed in the process. So if it comes to us and we don't have to do that step, that's a bonus for you and for us. So stripping, clipping, removing flower heads, picking berries are all fantastic ways of getting a collection. Please don't pull plants from the roots. It makes for a difficult to clean collection as well as it contributes to disturbance at the site. I love this photo. If you look closely, you can see the black specks under the yucca, the seed floating in the air. And this is a good example of how, use your creativity when collecting. They put a tarp below the yucca and someone's just shaking the seed out of the pods. To me, this is like they're guaranteed to get the majority of the mature seed. The undeveloped seeds probably still remaining in the pod. The next slide. So we just made a list of tools that might be useful for your collection. You can look through it, but I would also maybe add vacuums to this list. I know some people like to use vacuums, particularly for seed that gets carried on the wind really easily, so asters. I think it works great. Just be really careful to check a small sample and make sure it's not being damaged with that vacuum before you go ahead and collect the entire population. So In terms of caring for your seed collection, really, it's pretty intuitive. You can collect in a paper bag out in the field. You can collect in a bucket or a plastic bag. The important thing is that your seed is going to be dry, can dry easily. It doesn't take long sitting in a closed container for mold to grow. So maybe lay it out, dry it, and then transfer it to a paper bag. Avoid extreme hot, cold, wet conditions. Really, leaving your seed in the back of the truck or in a trunk in the hot of the summer is probably not a good idea. Also, make sure you have a label with a unique identifier on the outside of the bag as well as on the inside. We actually get very few seed lots confused or messed up, but this is a way, having that tag on the inside of the bag may seem redundant, but it's a way for us to double check and make sure that everything is where it should be. So at this point you might ask, what data do I need to collect at the site? And that's going to vary, depending on your project needs. But Kayla and I came up with this list of what we think is pretty basic, and you may add to it or not. Just be aware that if you're going to have your seed processed, their needs may be different than your needs, and that potential future users may have different needs than what you need. And we've included an example of our form, which is pretty basic, but there's some important fields on there that are necessary to us, as well as the Seeds of Success form, which is much more detailed, but we use this in lieu of our data form because it's got all the information that we need on it. And finally, how do I ship my seed collection? Please ensure that you include a data form, that the data form matches, that unique identifier is on the data form as well as the bag. Securely pack your seed in boxes, and that may mean different things to different people, but pad it really well, tape the bags. Some seeds escape really easily from bags. Ensure that your material is dry before you're sealing it. And if you have fleshy fruit, put it in Ziploc bags, maybe 2 Ziploc bags. Pack it in ice and for sure ship it overnight. So Kayla's going to continue on into the seed processing portion of the collection.
Yeah, thanks, Sarah. So talking a little bit about seed processing and testing and storage before we end, it's really important to remember that proper processing really begins as soon as you remove the seed from the plant. So all that temporary handling in the field and potentially in your office, like she just mentioned, all of that is important in taking care of your seed. So just talking a little bit about seed cleaning, is it necessary? One of the most important things to think about is, do I need to have my seed cleaned? It may not need to be cleaned if you're broadcasting it out on a site. Potentially you just need minimal cleaning for hydro seeding project. Or if you're sending it to a nursery or long-term storage where space is limited, it might really be necessary. So thinking about the challenges and the benefits of seed cleaning can help you answer this question. Is there a facility available? Can they meet my time frame? Especially in the fall at the Bend Seed Extractory, we're really busy. So it's important to make sure that we're going to be able to process your seed in the time that you need it to get it to a grower. What is the cost of cleaning or shipping, especially with fleshy fruits? Overnight shipments are not necessarily cheap, so something to consider. And then benefits, you know what you have. Your end product has seed tests that allow you to predict pure live seed. They aid in sowing requests, and it allows for nurseries to handle that seed with a little bit more ease because they know what they're getting. So what does the extractory provide? We extract, clean, and store seed for any governmental agency. If you ever have a question, please just give us a call or send us an email. We've processed over 2,700 different species, and we maintain detailed information about all of those collections. So like Sarah had mentioned, for your planning in terms of collection, if you are curious about a species that you might not be familiar with, please contact us or maybe another local extractory or processing facility, because all of that information can help you. And then know that we do arrange for a lot of storage and testing and shipping. Depending on the facility you're looking at, like the Bend Seed Extractory, we have the ability to work with small and large scale seed lots. So when you're focused on native plant collection, with a species that might have really tiny seed, we're able to handle a small handful of seed where potentially another facility might not. So when you're thinking about what does the extractory provide, make sure that the scale of your project is something that is well known to them. So thinking about seed processing, it can be very time consuming. Specialized cleaning equipment can be expensive. So just keep in mind that there are some low-tech methods available if cost or time is something that you don't have in terms of the extractory. So there are a lot of low-tech methods out there, but just know that we target seed here based on the physical properties of your target seed and then the properties of the non-target material. So whether that's inert or weedy seed, that's really what we're focused on in terms of processing and how we choose our machines and the methods that we're going to use. So here's a couple examples just to show you guys. This is Western needlegrass, and you can see that the picture on the left is what it looks like when it comes in, and the picture on the right is clean seed. So just keeping in mind that processing doesn't necessarily just mean removing inert material. In this case, it also includes removing that large awn that you can see. The same is true for bottlebrush squirrel tail. You can see that it is a little bit of a mess on the left there, but that's okay. You can see there are large stems, so that's material that we like to see. And then the photo on the right is the clean seed. So knowing that we're removing that awn and creating a flowable product for a nursery or a grower to use is really important. So what does processing look like? We wanted to share these photos with you so that you can see raw material, and then after we extract it from the flower head, there's a mixture of inert and seed, and then after that we do a finishing process which results in pure seed. And then you can see there's a photo on the lower right-hand corner that we just wanted to share with you. That's seed with inert material, and the inert material there is sand. So just thinking about some of the things that you might not normally, you know, flower parts, leaf parts, small stems, that might be intuitive. Knowing that some of those low-growing forbs and shrubs in windy, arid environments might be capturing sand within the flower heads is really important to note. We see a lot of sand here at the extractory, and it's sometimes very difficult to remove from your seed. So just thinking about when you're going to collect your seed, what you're really collecting. So talking a little bit about testing, after processing, it's really important to determine the quality of the seed by testing. It can help growers know the potential of the seed lot and also predict how much seed they might need to meet your target. And that's really the most important thing, is that we are all meeting your target for whatever your project is. That's what we want to do here at the extractory. We want to clean and process the seed to the level that you need it. And we want to provide the test that potentially your grower needs. So going back to that cycle I showed you at the beginning, we really are all focused on what your end goal is. So that's really important. So some of the testing we do here are seed weight and purity tests. And then we also do seed moisture and 100% extra, or 100 seed X-ray. By taking those tests, that allows you to calculate your pure live seeds that you might need for a grower. It's also important to take into account viability. We don't do viability testing here at the Bend Seed Extractory, but we do provide a transfer for that, the ability for it to be done. Viability testing for us is done at a certified seed lab, and it's really important to understand what viability is. It's the estimate for the potential for seed to germinate. So thinking about the two tests that can be done, germination and a tetrazolium test, those are your options. And a lot of people ask, why would you choose one over the other? A couple of things to consider are the amount of seed, the timing of the results, maybe uncommon species. The TZ is going to estimate the potential for seed to germinate, where the germination test actually measures the actual germination percentage and the rate, because it's measured on a daily account. So that's an important thing to think about. So in testing in general, just remember that tests really allow for sowing rates to be calculated, and it results in the efficient use of your seed. So knowing how much time and effort you put into this collection, we really want to help you use your seed to the most efficient way possible. And tests really do contribute to that. So how is seed packaged? Here at Bend, we package in 6 mil plastic heat-sealed bags to prevent moisture transfer in or out. This is important because we really want to maintain viability. It's really beneficial to have storage available if your desired species has irregular seed production or low yields from year to year, so that you can capture variability in your population to create a strong restoration project. And sometimes that may result from multiple collections year after year and then combining those. So really think about, why is storage important, and how can I use storage as a tool to benefit my project? What are some storage options? They vary by facility. Freezer storage is usually best for long-term storage, and just remember, like Sarah mentioned, some species are inherently short-lived and cannot be stored for long periods of time, especially like the acers. So really think about, what are my options for storage, where am I going to store it, and then, of course, thinking about your planning, your whole long-term goal, what about storage fees, shipping fees, service charges, to really think about, how am I going to accomplish this restoration project. So that concludes our talk. Thank you all for attending, and if you have any questions, please type them in so Janae can ask them.
Great. Thank you so much, both of you. All right. The first question is from Catherine Beal. How many years is it okay to collect at the same site, and how many years should we use the same collection for grow out if still getting good yield?
Ooh, that's a good question. Unfortunately, I'm going to answer it with probably not everybody's favorite answer. It depends. It really depends on the species, and it really depends on where you're growing it out. A lot of these species, depending on your population, you can go back to them year after year without a problem if you're collecting only 10 to 20% of the actual produced seed on the site. We do like to kind of tell people ethically it's nice to not visit the site every year and really think about potentially a 3 to 5 year return cycle. However, that's not always realistic, because a lot of projects are really dependent on certain seed, and populations oftentimes that are good producers and nice sites may end up having that visitation a few years in a row. So do keep in mind that. And then if you are collecting seed and you would like it to be grown out, we see folks use the same collection for multiple years. And I would say, again, the same time frame, like 3 to 5 years. But that seems to be, that's when they start to exhaust that collection.
Great, thank you. Yeah. Now, Kim Freimeyer asks, where can we get the information on yield data?
Kim, you can contact either I or Kayla at the Bend Seed Extractory, and I would suggest starting with us. We, I think, probably have the most wildland collections to report about. If we do not have your species, you could maybe refer to a similar species that we do have data for, and just plan on over-collecting until some more data is available. You could try researching other resources. You never know what's out there. But please contact us either through email or phone, and we'd be happy to give you whatever we have.
And if we don't have it, you know, some of these key species that have become restoration concern, we may know where to direct you. So, yeah.
Wonderful, thanks. Okay, Eric Freeman has a few questions in a row. The first is, what is meant by the average yield column on the collection amount tables for grass, forbs, and shrubs that you showed?
So that's just, all that is really basic. The average of all the collections for that species, the average of we record all the weight as it's received, so the raw weight, and then we record the weight of the clean seed. So that column is just the weight of the clean seed divided by the weight of the raw material. It does not take into account how much of that seed is viable or filled. That would be, if you wanted that information, I would go to the PLS per pound of raw weight material. That column is really intended to give you an idea of, because a pound is a pretty easy number to estimate in the field. If you're collecting a pound of Wyoming big sage, know that PLS per pound column, that's close to what you're going to get as a final product.
Thank you.
Does that answer?
Yeah. All right. How early in the seed development process can I collect sagebrush seed? Do I need to wait until it is fully dried and falling out on its own, or can I collect slightly before then, and will it finish on its own?
That's a question everyone has. Seed does not after ripen. That's a really commonly misunderstood term. After ripening is actually a term that's used for conifers, and it's a metabolic process that happens after collection to allow for germination. After ripening has nothing to do with seed development or maturity. So if you collect seed that is immature, it immediately stops maturing when it is removed from the plant. So if you collect seed immature, you may find that a few of those will germinate, but they're going to have very low vigor, and you're going to end up with a poor quality in terms of any sort of storage. So you really need to make sure that the seed is fully mature. Something like sagebrush in particular actually does hold on a little longer than some of those species that are in a pod, like Sarah was mentioning. But we oftentimes see the best sagebrush collections happening in late October, November, and December. So really waiting till the last possible maturity date is great. And another part to be able to capture some of that in capturing phenology is really kind of making multiple collections of maturity. Not every plant is going to mature the same. So if you're trying to do some early collections, just make sure that you're also capturing some of those late bloomers as well. And if you have some specific questions about your particular sagebrush population, please just give us a call. We'd be happy to talk to you about it.
Very good to know. Thanks. And do most collectors collect sagebrush by stripping or by pruning seed stalks?
I would say it's about 50-50. We've made a huge attempt to try to encourage people just to strip it, and I think our collections are getting more on that end than cutting a branch. Like I was saying earlier, if we receive branches over 6 inches in length, we're forced to kind of be creative and come up with a way to get that seed off of the branch before we can even start to process it. So please, please, yes. And Kayla's made a little note here that another thing to consider is that when we have to do that, when we have to come up with creative ways and time-consuming ways of getting seed off the stem, there's often going to be an increased cost to us, and that'll be passed along to you. So I would say strip sagebrush seed, shake it. Even shaking works really well. Just tipping the seed head down into a bucket and kind of gently shaking it. The seed that's mature almost always will fall off, and then you can revisit that site, that plant again, and do the same thing maybe in a couple weeks. I've also seen collections that have been vacuumed of sagebrush, and honestly it seemed to work pretty well. There were a few damaged seeds, but they ended up meeting their target with no problem by using I think it was a leaf blower in reverse.
Wow.
Yeah.
Okay, thanks. Alyssa Salmore asks, does the Bend Seed Extractory work with federal government agencies only, or do you have the ability to contract with state agencies to process seed?
Oh yeah, so we are any governmental. We work with a lot of cities and counties, state entities. We can also work with universities that are affiliated with the state. So we've done some work with various universities in terms of research, any sort of plant materials center. The only thing is we are not allowed to compete with private industry. So private individuals we are not allowed to clean seed for. However, if you were to contract out your seed collection as a state entity, then we are allowed to clean it. We just cannot accept private money. But we can accept anything from a state agency. Yes.
Great, thanks. Kelly Evans asks, how long does a germination test take and how many ounces of seed does it require?
That's a good question. Again, it depends, unfortunately. A germination test requires 400 seed. It's not based on grams, it's based on seed. So it takes 400 seeds, and depending on the species is how long it's going to take. Some of the shortest germination tests are your grasses. We can see the results for those in just a few weeks. Something like 9 bark is going to be closer to 8 weeks. And some of your fleshy fruits, like the berries or conifers, can potentially take up to 120 days. So there's a lot of variability. If you have any question about a specific species, I'm happy to answer those questions just by a phone call or email.
Great, thanks. Larry Loftus asks, how much seed of Poa secunda would need to be collected for a grower to grow it for increase?
I think you'd have to to be safe, I would contact the grower and make sure that they can give you a good number. I'm going to say probably not less than a pound is safe.
Okay, great, thanks. Holly, I'm going to hope I say your name right. Sletaland? I'm sure I did not say that right. I'm sorry, Holly. She asks, I had been told that if you cut the stem and let it dry with the seed pod, especially annuals, that the seed would continue to mature. But am I understanding that that is not the case?
The best answer is that is not the case. There are a lot of rumors out there, and some people who are really familiar with particular species and have a lot of notes and have taken a lot of data might know timeframes, but from what we see in general collection, for a few thousand different species, a lot of people who think that they are clipping it and then allowing it to after ripen almost inevitably end up with immature seed. So unless you are really familiar with your species on a time frame, and I think that most species it's a matter of hours, you're not going to have strong genetics in your population. You're going to end up with a very weak seed. It's not going to store long, and like I said, it's going to have low viability. So that is not really a tool that we recommend at all.
Okay, thank you. Weston Miller asks, when testing seeds, do they go through a stratification process?
That's a good question. If you are having a germination test done, yes, there will be a standard stratification process, and it depends on the species. The important thing to remember that with a lot of these wildland collections that are native species, we don't necessarily know the best stratification process. So it's really important to understand that if you get a germination test back that maybe isn't as high as you expected, to know that the standard protocol is coming from the Association of Seed Analysts, and it's what they have established to use for testing. It doesn't necessarily mean it was the best method. So yes, it does happen. Some species we know a lot more than others.
Great, thank you. That looks like the last question. There was a comment that this was a great webinar, and thank you. Yeah, that was the last question. We would greatly appreciate it if you would take our 3-question survey of this webinar that will appear after the webinar has ended. A recording of this webinar will be sent to you automatically through the GoToWebinar system tomorrow, and I will also post the webinar along with the PowerPoint presentation on our website at the Great Basin Fire Science Exchange within the next couple of weeks. The third webinar in this series, titled Procurement and Application of Native Plant Material in the BLM, will be presented tomorrow at 11:30 Pacific, 12:30 Mountain, in case you're interested. And if you have more questions regarding this webinar, please feel free to email Kayla and Sarah. If you have requests for future webinars or questions for me, please email or call me at any time. Thank you all for your presentation. Wait, you know what? I just see one more question. We might as well answer it since we're still on. Okay, someone said, I joined the webinar late, so please disregard if it has already been asked. On the Bend Seed Extractory website, there is a page with processed seed reports ending in 2012. Will Bend continue to provide these to BLM?
Yes. I'm guessing that this is coming from someone with the CV Success program. So an unfortunate thing is our website is internal only. It's not available for the public. So sometimes, depending on the website, it's not updated. I know there's a few misguided websites out there, which I apologize. I'm not really familiar with all of them. But all of the Seeds of Success information for the BLM actually goes through Megan Haydett at the national office. So she has the most updated of those. If you're curious in yours in particular, just please contact me or Sarah. We can run inventories and reports fairly quickly for you. So the Forest Service inventories do get updated on our website every 6 months. But because the SOS program is so different and it's paid out of the Washington office, everything goes through that. So if you have a specific question about inventory or reports, please just contact us and we'd be happy to help you.
Great, thank you. And one final comment, Brad St. Clair said, great job, Kayla and Sarah. Useful information. Thank you. So thank you very much to both of you for your presentation.
Thank you.
All right, and thanks everyone for participating. Have a great day, everybody.